Hello guys, welcome to my YouTube channel. So today we are going to see some specific problems on biochemistry and molecular biology. So let us see what our first problem says. It says that E. coli is a rod-shaped bacterium with 2 micrometer length and 1 micrometer diameter. So this means this is the length L and here we have 1 micrometer diameter. So from here we can calculate the radius R. It also says that the average density of a cell is 1.28 grams per ml. So this is the density of the cell. So if we designate it by rho, then rho is equal to 1.28 grams per ml. Now they are saying that approximately 13.5% of the weight of E. coli is soluble protein. So that means you have to calculate the total weight and then you have to take 13.5% of that total weight. Now you are given length, radius and density. So how will you calculate the total weight? First you calculate the volume. If you take this to be a rod-shaped bacterium, that means it is a cylinder. If you take this to be a cylinder, then the volume will be pi r square L. So this will be the volume, and the weight, that means the mass, what will it be? It will be volume multiplied by the density rho. So you have the rho density given, you can calculate volume and from there you can calculate the total weight W, and then you take 13.5% of this total weight, you will get the weight of the specific soluble protein in E. coli. Alright, so out of the total weight of E. coli cell, 13.5% of that is what the weight of the soluble protein present in E. coli is. Now they are saying that calculate the number of molecules present in a particular enzyme per cell if the enzyme has a molecular weight of 100,000 and represents 0.1% of the total soluble protein. That means what? Out of the total weight of E. coli cell, 13.5% of the total weight is this soluble protein, and out of that the 13.5%, 0.1% is the total soluble protein. So I will make it simple when we will do the calculation. And one thing, if you remember, how will you calculate the number of molecules? Number of molecules or the number of moles is calculated as to be as n by Na, where n is the number of molecules and Na is the Avogadro number. So what will be the value of this n? It will be number of moles multiplied by the Avogadro number. Alright, so these are the formulas that you need to do, that you need while solving this sum. So first let us calculate the volume. Let us proceed step by step. So here, as you can see, first we have calculated the volume. This is the radius, 0.5 micrometer, because the diameter was 1 micrometer. So it will be half of that, and we have calculated the radius in centimeter and the length also in centimeter. So the volume comes out to be 1.57 10 to the power of minus 12 centimeter cube. So this is the volume. This is the first step of calculation. Now let us proceed to the second step of calculation, which is the calculation of the total weight of E. coli cell. So how will we proceed? From volume, we will multiply volume with density to get the weight. So here, as you can see, this is the second step where we have multiplied volume with density to give the total weight. So this is the second step from and the weight comes out to be around 2 into 10 to the power minus 12 gram. Alright, now this is the total weight of a single E. coli cell. What have they told us? They have told that out of this total weight, 13.5% of this total weight is the weight of this soluble protein. So that means what? You have to take 13.5% of this total weight, which will give you the weight of the soluble protein. Alright, and out of this 13.5% weight of the soluble protein, 0.1% of that 13.5% is the weight of the enzyme. So if you calculate the amount of enzyme, it means 13.5% into 0.1% into 2 into 10 to the power minus 12. So this comes to be 2.7 10 to the power of minus 16 grams. So this is the amount of the enzyme. I will make it simpler. So what was our total weight? This was our total weight, 2 into 10 to the power of minus 12 gram. All right, this was our total weight that we have calculated. They have told us that 13.5% is the 13.5% of the total weight is the weight of the soluble protein. So you multiply it by 13.5%. all right, now the thing that you are getting, this is the weight of the soluble protein, and 0.1% of this weight that you are getting, 0.1% of this entire thing is the weight of the enzyme. So it means 0.1% multiplied by this entire thing is the weight of the enzyme. So if you multiply this whole thing with 0.1%, so that is what they have done here. All right, and the answer comes out to be 2.7 10 to the power of minus 6 gram. Now how will you calculate the number of molecules? I have already shown you. You can multiply Avogadro number with the number of moles. So here, this is now how do you calculate number of moles? Weight by molecular weight. So this is nothing but the number of moles. So here weight is 2.7 10 to the power minus 16 into 6.023 10 to the power 23 divided by 10 to the power 5. All right, 10 to the power 5, this is already given to you. This is the molecular weight. This means 10 to the power 5. So the number of molecules comes out to be 1626 molecules per cell. All right, so this is the number of molecules per cell present in E. coli.